Can you all now see my screen? Is my screen visible?
Yes.
Yes, miss.
Fine, let's start. Okay, paper 2 is 6 questions. It is going to be covered in 1 hour and 15 minutes, and it is a relatively long paper, by the way. The 6 questions, I think they need an extra 10 minutes. So while you're solving paper 2, put this in mind that you have limited time. Okay, now let's start. Number 1. The malarial pathogen Plasmodium falciparum enters red blood cells after a person becomes infected. After some time, each cell of the Plasmodium falciparum divides to form daughter cells. Figure 1.1 shows a cell of the P falciparum that is forming many daughter cells. Cell surface membrane of red blood cell. This should be a red blood cell. With reference to the figure, suggest how the presence of the P falciparum affects the red blood cell. 3 marks. So can you give me 3 changes that happened to the red blood cell? How did the presence of this pathogen inside the red blood cell change its shape?
The red blood cell became more swollen, and the cell surface membrane is irregular in shape. It's not a disc shape.
Yes, of course, we know very well that the red blood cell should be small and biconcave. It is not biconcave anymore. The cell membrane became irregular. It's no longer not biconcave. Can you think of something else? How do I know that we need 3 points? Because it's out of 3 marks. So you need 3 changes. What else can you say happened to the red blood cell? You have to think of 2 other changes.
It would burst because
It is bursting, correct. You can see that it is bursting. Because what?
when the pathogen does its asexual reproduction in the liver, it then goes to the red blood cell and then it bursts so that it can spread the toxins.
Yes, the pathogen, they said in the question that it is forming many daughter cells. So it is multiplying inside the red blood cell. It will make it larger and it will burst. Actually, writing that it would make it larger, it's very larger than normal. This is another change. Think of a third change. Think of a fourth change. Boys, what do you see inside the red blood cell? Do you see anything or it is empty? It only has the pathogen. Speak up.
It only has the pathogen.
Okay, what should it have and you don't see it? What is the only thing that is found inside the red blood thing and inside the red blood cell anyway?
Hemoglobin.
Yes, I don't think that you see any hemoglobin. There is no hemoglobin. Okay, the pathogen consumed everything, and the pathogen is multiplying and it is making it larger than normal. It is no longer biconcave, like Mariam said. It is irregular or it has an irregular cell membrane. It's larger. And do you think it's going to be efficient in carrying oxygen in its normal function?
No.
No, so it will no longer carry oxygen. So it will not carry oxygen. It will be destroyed. The red blood cell will be completely destroyed. It will change in terms of shape and in terms of function, and it's not going to carry out its function. Okay. So this question has got nothing to do with what you studied in the syllabus. It depends on your past information about red blood cells. When they tell you with reference, then you will refer to the figure and you will have to get the most information out of this figure. And how much information you need depends on the number of marks. This is 3 marks, then you will write 3 changes, whatever 3 changes that you see. It's an easy question, by the way, but you usually do not notice the number of changes that you have to write. When the P falciparum divides, there is unequal division of the cytoplasm to form small, genetically identical daughter cells. Outline the events that occur in the cell of the P falciparum to form the daughter cells shown in the figure. So what are they talking about? Which events do they want? Outline the events that occur in the cell of the P falciparum to form the daughter cells. So from the moment it starts until it divides and give me the daughter cells. Mm. How will you
Mitosis
answer?
will-
Yes, they want you to talk about mitosis, actually, the events that happen in mitosis. But not only mitosis, even before that, a little bit, the whole
الانترفيز.
cell cycle. Yes. Okay. So the events that happen is the cell cycle that happen in any cell until it divides. And then they specifically talked about the division of the cytoplasm. So this is which event? Which step?
Cytokinesis.
Cytokinesis. Maybe this you will discuss in detail and the rest you will outline. So if you will talk about the whole cell cycle, what will you say? The cell cycle starts with what?
Interface.
Yes, it starts in interphase. And what happens in interphase? They want
Growth
an
بعد
outline.
كده growth.
Yes. Which growth? G1.
G1, yes.
And then?
Growth of the cells. Then S phase, they replicate the DNA.
Yes.
And
Always
then
when you discuss, wait, always when you refer to replication of DNA, always use
semi-conservative
the term
replication.
semi-conservative replication of DNA. Okay. And then you have the,
G2.
yes.
More growth and errors are checked.
Very good.
Uh,
And then,
and then mitosis.
okay, what are the steps of mitosis?
Prophase,
Mm,
metaphase ولا
mm,
أو
mm.
anaphase
Yes,
وtelophase.
yes. And you can write a sentence, just a sentence, because they clearly said outline, sentence about every step. And then what will you discuss in detail?
الcytokinesis.
Cytokinesis, yes, because they mentioned it. And the cytokinesis, how will you describe it?
The cytoplasm um, shrinks from the middle of the cell
Okay, we
until it
don't use the word shrinks. What do we use? What does it form? It contracts the cell membrane, it shrinks, okay. It forms a cleavage furrow. It will pinch off. It's like it will squeeze from the middle.
uh,
And the cytokinesis in animal cells is different from the cytokinesis of the plant cells. And the cell will divide unequally in that case because they gave you this part until it will form identical daughter cells. Okay?
okay.
Now, in 2013, the World Health Organization set a target for researchers to create a vaccine for malaria. They love talking about vaccines. The WHO required the vaccine to show 70%, 75% efficacy and be ready for use by 2030. Efficacy is a measure of the effectiveness of a vaccine in reducing the number of new cases. A trial of this vaccine in Burkina Faso in 2020 achieved a 77% efficacy over a 12-month period. A control group received a vaccine for rabies. Vaccines stimulate an immune response with the production of antibodies. Okay, C. Explain how antibodies will reduce the spread of the malarial pathogen through the bloodstream. Now, this is a direct question. How do the antibodies work? This is a direct question.
They
Huh.
bind to the antigen.
Okay. No, there's a
Uh,
list of things. Remember, I know ball? Remember?
yes.
Okay. I for what? Quickly. Your exam is in 2 weeks. يا رب. What is I? Others speak up. Adham, Maryam. What's I? What's N? What's O?
I is
What?
immobilization.
Yes. What will you say in immobilization?
It prevents it prevents the pathogen from moving, the bacteria or
Yes,
pathogen.
it attaches to the flagella of the pathogen. It doesn't spread. It doesn't move. What is N? What?
Neutralization.
Neutralization. It will neutralize the toxins. It's antitoxins. What is O?
Opsonization.
This is the most important. What is opsonization?
It marks the I think it marks the pathogen for the phagocytes.
Yes. Of course, this is the pathogen, this is the antibody, and the phagocyte will have a complementary what?
Receptor.
Receptor that will join with the antibody. The pathogen cannot see the pathogen except if it is marked with an antibody. Okay, so it helps the phagocytosis process. What is B?
Block.
It will block it. It will prevent it from entering the host cells. What is A?
Agglutination.
Yes, it will make the pathogen stick together. What is L?
Lysis.
Lysis. It will make the pathogen rupture. And of course, you have to mention that the antibody is specific. It binds with a specific antigen. And when the antibody binds with a specific antigen, this forms which complex? Just like the enzymes.
Antigen antibody complex.
Antibody antigen complex. Now, why do I feel that you know the information, but you always need guidance as from where to start? This is for everyone. You all know the information, but it's just like you are confused. You don't know from where to start. This needs more practice because the questions, unfortunately, are not straightforward. They are twisted questions. Okay, so here you will discuss antibodies and you have a lot of things to say. You can even discuss the structure of the antibody because you also studied that. The variable region that was specific to the epitope and the constant region that binds to the receptors on the phagocytes. You have a lot of things to say in this question. It's an easy question. Some vaccination programs have been more successful than others. Discuss the factors that contribute to the success of a vaccination program. Again, this is a direct and easy question. You have lots of reasons why a vaccine will be successful. Kareem, can you tell me why a vaccination program will be successful?
Because many people will vaccinate herd immunity.
Excellent, because the people are willing to become vaccinated, and when most of the population is vaccinated, you will provide herd immunity. First, Kareem, I want you to tell me how many points will you write at least in this question?
5.
Correct. So give me a second reason why the vaccine will be successful. It's a very easy question.
Because the vaccination is a dead or a weakened pathogen. when يعني البادي حامل immunity against it فهيكون عنده memory cells عشان لو الباثوجين جه تاني ممكن immunity system يقاتله يعني.
Okay, you're right. It will stimulate active immunity and create memory cells, which will provide lifelong immunity. But any type of vaccine will do that. You mean a particular vaccine that is stronger than the other? Which vaccine is stronger than the other, the live vaccine or the dead vaccine?
A live vaccine.
Yes, maybe this vaccine is a live vaccine. You have a live pathogen, but it is weakened. Okay, so this is the reason. Give me a third reason why it will be successful. It is a strong vaccine. It's more effective. Hmm? Give me another reason.
No mutation in the gene.
Excellent. Of course, the pathogen does not mutate. No mutation in the pathogen. The pathogen does not show antigenic variation, antigenic shifting, antigenic changing, the antigenic hiding or antigenic concealment. The antigen is stable. This is a very important reason. If it was not stable, I cannot make a vaccine. Give me a fourth reason and a fifth reason.
ممكن أسأل عشان يعني لو ما فيش بوستر يعني ما فيش بوستر دوز يكون
Exactly,
أقوى
of
يعني.
course. No booster dose is needed, so it's easy. Only one dose is needed, so this will make it more successful. Another reason, it's
The
expensive
vaccine
or
is
cheap.
thermostable.
Of course, it is easy to store. Easy to store. It is thermostable. It is freeze-dried. Remember freeze-dried? Thermostable. It is cheap. Okay, it is easy to deliver. There is cooperation from the government. People are educated. People want to be vaccinated. And Corona, at times of Corona, the government bought lots of vaccines from China and they were thrown in the River Nile. Do you know that? Because the people did not want to get vaccinated. So the people were not cooperative. So the vaccination was not successful. Any one of those. This is actually a very easy question. Okay, we finished question 1. Glucose is used in the synthesis of amylose. Glucose is first converted to glucose 1-phosphate, and then starch phosphorylase enzyme is an intracellular enzyme that can catalyze the synthesis of amylose from G1-phosphate, which is the substrate for the reaction. I want to stop here. What is amylose? Amylose is what? Protein or starch or fat? Remember, what is amylose?
Protein.
حرام عليك يا نور. Amylose and amylopectin. This is what?
يبقى lipid. لا ثانية، مش ده
Amylose and
sugar.
amylopectin.
أيوه
Yes,
carbohydrate صح.
starch.
أيوه أيوه.
Starch. The amylose is the coiled part of the starch, not the branching part. It's starch. It's important to remember that because they will tell you that there will be color changes with iodine. Okay, let's look here. This is the glucose 1-phosphate, simply glucose. In the presence of the starch phosphorylase enzyme, it will be converted to starch. Amylose plus phosphates, part of the starch and phosphate ions. Students used a calorimeter to investigate the progress of the reaction. The students made a reaction mixture containing 0.01 mole per decimeter cubed of the glucose in a buffer solution at pH 6, and a very small amount of amylose was added to initiate the reaction. And then a solution of starch phosphorylase was added to the reaction mixture, and samples were taken out at 1-minute intervals. Each sample was added to a dilute iodine solution, stirred, and then poured into a cuvette. And the absorbency of each solution was recorded. Now, this is one of the rare exams that talks about the calorimeter in detail. What is the absorbency? The results of the investigation are shown. And you have here the time after addition of the enzyme and the absorbency. And the absorbency is increasing. Now, we have a graph like this in your notes, and we talked about absorbency in a calorimeter. What is the absorbency? It is the measure of what? How much is what?
How much light is absorbed by the color?
Excellent. Excellent. How much light is absorbed by the machine? And the darker the color, the more the...
The absorbance.
Excellent. So now, why is the color of the iodine getting darker? Because what is being produced as the reaction is progressing?
Amylose.
Very good, which is starch. Okay, it's a very simple, by the way, experiment, but they have to confuse you. This is their job. They are simply telling you that we have glucose, and this glucose is converted into starch. It is as simple as that with the help of an enzyme. And every time, or the more starch is produced, the darker the color and the more the absorbency. Here we created a calibration curve. It's not really calibration because it's not over concentration, but here is the absorbency over the time. Boys, do you understand the graph? Do you understand the experiment?
Yes.
Good. Explain why the absorbency then, Noor, increases as shown. Why would the absorbency increase? We just said it.
Because the more starch is يعني الأيودين breaks more
Yes.
starch.
No, the iodine doesn't break more starch يا حبيبي. The iodine detects the presence of starch.
قصدي بيخليه darker.
Okay, yes. So the more the starch, the darker the color. That is why it's going to show me or give me a higher absorbency. The absorbency is the number on the calorimeter machine. The more the machine is absorbing light, the greater the number. And the darker the color, the more the absorbency. Do you all understand that? I hope so. The students took their final sample at 12 minutes. Predict the results for the absorbency if the students had continued to take samples for a further 10 minutes, and explain your answer. Now, this is a tricky question. At 12 minutes, the absorbency was very high. The absorbency was its maximum. What do you think will happen to the absorbency if we waited more time? Will it keep on increasing? Because there are only two options. It will increase or it will remain constant. What do you think will happen? Who do I have here? Farouk, you're not speaking. I did not hear your voice. What do you think will happen to the absorbency if I keep the reaction longer and longer?
Uh,
The color will become darker or it will stay constant?
this will become darker.
Why will it become darker? We have glucose converting to starch. Every time it will convert to starch, it will make the iodine darker. Will the glucose keep on converting to starch forever?
No. Okay.
So what's your prediction?
It will stay the same color.
Yes, it will become constant. It will reach a plateau. The color will remain constant. It will stay the same, or it might increase, but very slowly. Now, what is your explanation?
Because all the starch has already been changed. It's already all been changed. There's nothing left to be broken down.
Because all the glucose have been converted to.
The starch.
Correct. Now, in an graph, remember the enzyme graphs that we took and the limiting factors? Here, if we have a rate of reaction and the progress of the reaction or with time, and the reaction increases and then it becomes constant. What is the limiting factor in this case when all the glucose, all the substrate has been converted into product?
The glucose.
Yes, the glucose, it is now a limiting factor. Here is the substrate concentration on the on the x-axis. So all the glucose has been converted to starch. And then when all the glucose is finished, the reaction will remain constant. Why? Because now the glucose, okay, or the substrate is the limiting factor. The substrate is, no, not the substrate is increasing. Here, let's say any other factor here. Let's put time, like we did. Okay, and they're telling you in the information that the glucose is constant. When all the glucose is finished and converted to starch, there will be no more substrate to work on, and this will be my limiting factor. So the explanation, you can simply say, because all the substrate has been used up, you will take the grade even if you don't mention the limiting factor. But the substrate concentration here is the limiting factor. Clear? You all understand this question?
Yes.
Okay. In my opinion, this is a little bit tricky. So let me highlight it so that if you revise this exam, state an advantage of using a calorimeter in determining the progress of the reaction.
It's a quantitative result, not a qualitative result.
Of course, it's a quantitative. Now, this can be enough, but quantitative means what?
It gives numerical results or with
Yes,
numbers.
it's a number, which is more accurate, of course. There is no human error here. You cannot here argue with that. The reaction catalyzed by starch phosphorylase occurs at the end of the amylose molecules. Describe the sequence of events that occurs when starch phosphorylase catalyzes the addition of a molecule of glucose to the end of an amylose molecule. This is 4 marks. How will you answer this question? Describe the sequence of events that occurs when the enzyme catalyzes the addition of a molecule of glucose to the end of the amylose molecule. I will not give you the answer. You will give me. What are they asking about? Noor, what will you say?
I can't understand the question.
Yes, this is the problem. Even though the question is very simple, the questions are twisted and they confuse you. They intentionally confuse you. Here, they are simply asking you, how will the enzyme work? What is the mode of action of the enzyme? That's it. That's it.
And it has an active site complementary to the substrate.
Exactly. Can you imagine that this is the answer? And you will discuss the induced fit. Okay. So they are here. This is a very easy question, but they intentionally want you to ask yourself, I don't know what they want, but they will But when you are confused, just read the question more than once and hopefully you will figure out what they want. They want the sequence of events that occurs when the enzyme, this is an enzyme, will do the catalysis reaction. So this is the mode of action. You will mention that the enzyme has a specific active site. It is specific, okay, the specificity of the enzyme. It will bind with a complementary substrate. And when it does, this is, you can discuss the induced fit. You can say that the active site will move a little bit so that it is complementary to the substrate. When it does, what does it form?
Enzyme substrate complex.
Very good, the enzyme substrate complex. And then it will give me the products. Now, what if I am still not sure if this is what they want? Here, they are talking also about the addition of a glucose molecule to the end of the amylose. Now, remind me, this amylose is a polymer or it is made up of what? We studied it in detail in biological molecules.
It's a polymer.
فاروق. It's a polymer. Yes, يا حبيبي يا نور ذاكر شوية. It's a polymer made up of what? What is the monomer? مريم فاروق Kareem. Amylose is a polymer made up from what? Which monomer?
Alpha glucose.
Yes, alpha glucose. You have an alpha glucose bonded to an alpha glucose. What is the bond? What's the name of the bond? This is carbon 1. Huh?
Glycosidic bond.
Yes. So the enzyme will catalyze the making of the glycosidic bond between the alpha glucose monomers. And this bond has a name, a specific name, between carbon number what and number carbon what?
104.
Yes, 1 and 4. So the enzyme will catalyze the making of the bond. This is alpha hydrogen above, OH below, and the same here. And this is a condensation reaction. You took it. So the enzyme, the mode of action, induced fit and complementary active site and enzyme substrate complex and making the product. And it will create the bond in the amylose, which is the alpha 1 to 4 glycosidic bond. Also, you can add something. How does the enzyme catalyze reactions? What do they do? Which energy do they reduce? They decrease which energy to catalyze any reaction, whether this one or any other reaction? Give me an answer. They decrease an energy that starts with the letter A.
ATP.
Excuse me?
Activation energy.
Yes,
Activation.
they decrease the activation energy. Because when you studied the mode of action of enzymes, you studied that they have either key and lock, which is the old method, or the induced fit. The active site changes a little bit so that it fits around the substrate. It is complementary, it is specific, it involves an enzyme substrate complex, etc., etc. And you also studied how the enzymes generally catalyze reactions by reducing the activation energy. Remember, they make the substrate unstable. They pull the substrate out of shape. They change the charges. They find an alternative pathway. They move around the charges or the polar groups in the substrate so that it is unstable, so that it reacts because the substrate is stable. We want it unstable. So when you have a question like this and you don't know exactly what they want, I keep on telling you, write everything you know. Write everything you know to be on the safe side. You will discuss the mode of action of enzymes, and you studied two mode of actions. You have the induced fit and the lowering of the activation energy. And they even asked you about a particular bond that you already know, so you can discuss it at least briefly, because this might be what they want. Do you understand? Are you all with me? Because they are being smart. You be smarter. They are asking you a tricky question, not a straightforward question. Okay, I don't know exactly what they want. It's okay. I'll write everything I know. This is what I'm trying to tell you. Write everything you know about this topic because you don't know exactly what they want. Let's look at the mark scheme. Where is the mark scheme? This is number 2, I guess. here. Okay. I was solving it before. It will bind with an active site. It will form enzyme substrate complex. It will add to the ends of the amylose. It will bind to the active site, enzyme substrate complex again. The active site will change in shape. We said induced fit. It will decrease enzyme activity. It will form the alpha 1 to 4 glycosidic bond. Okay. And any detail of binding to the active site, even this is mentioned. Interaction between the R groups or the formation of hydrogen bonds. So in this small part, they mentioned everything. They mentioned the mode of action, the induced fit, and the reduction of activation energy, and the name of the bond. You know what? You can even draw a diagram. Accept a diagram to show the bond formation. Even this is accepted. Okay? So, by the way, I was also solving it with a bunch of students, and they do the same mistakes, and they are also confused. So you're not different from anyone else. They are intentionally trying to confuse you. Don't be surprised and don't be upset. They are trying to confuse you. Okay? Now, where are we? I hope you're all with me. Now, 3, we are halfway through the exam. Hemoglobin is a complex protein molecule made up of 4 separate subunits, as shown in figure 3.1. So this is the hemoglobin molecule. The shading represents the 2 different polypeptides that form a molecule of hemoglobin. State the names of the 2 different polypeptides. This hemoglobin is made up of how many subunits? You already know it. What are the polypeptides that make it up?
Alpha and beta globin.
Yes, alpha globin and beta globin. I don't have to write it down. Identify the structured label P in the figure. P. Now, again, this is confusing. Is this one of the polypeptide chains? The polypeptide chains, either it's coiled or
No, it's not.
beta. It's not. So what is it? What is present in the hemoglobin that is not an amino acid, that is not a protein? It's the what? It's in the center here. You have 4 of them.
Iron core.
Yes, it is the heme group. It is the heme group or the iron group that holds the hemoglobin, that holds the oxygen. Figure 3.1 shows some of the levels of protein structure. State the level of protein structure that is not shown.
Primary.
Yes, you do not know the sequence of amino acids. Correct. Now this is easy. Hemoglobin is involved in the transport of carbon dioxide. Molecules of carbon dioxide enter red blood cells as the cells travel in capillaries through muscle tissue. Some of these molecules are converted into carbonic acid. Explain how hemoglobin is involved in the transport of carbon dioxide molecules that are not converted to carbonic acid. Now, this question is not asking you about the common equation that we know. The carbon dioxide reacts with water. How will the hemoglobin carry the carbon dioxide if the carbon dioxide does not react with water? Remember, we said the carbon dioxide is either transported in the plasma or in the red blood cell. And in each situation, it either reacts with water or it does not react with water. Remember this diagram? In the red blood cell, it reacts with water or it does not react with water. If it reacts with water, then it will be carbon dioxide plus water, the carbonic acid, and it will dissociate and so on. But this is not what they want. They want to know what happens to the carbon dioxide if it does not react with water. What will happen to the carbon dioxide?
Will react with hemoglobin.
Yes, it will react with hemoglobin. The hemoglobin will react with the carbon dioxide to give me what?
Carboxy hemoglobin.
Carbamino hemoglobin.
Carbamino. أيوة.
Hemoglobin CO2 is carbamino hemoglobin. Now
When
I'm
does
happy that
طيب carbo التانية when does it form? Carboxy hemoglobin.
carboxy hemoglobin, this is when the hemoglobin binds with the carbon monoxide
Carbon monoxide. Okay.
to form carboxy hemoglobin. I am very happy that we are all together now because I'm going to revise the hemoglobin again. The hemoglobin is very important in your syllabus. It is a globular protein. made up of more than one polypeptide chain working together as one unit. The polypeptide chains are 2 alpha globins and 2 beta globins. I think you know this, with a central prosthetic group that will bind with the oxygen. Now the central prosthetic group, this group, it binds with the oxygen, but it also has a high affinity to another molecule. What is it, Noor?
Carbon monoxide.
Correct. So this central iron group or heme group or prosthetic group, it binds with oxygen, but it actually has a higher affinity to carbon monoxide. When the hemoglobin binds with carbon monoxide, what is the product produced? Hemoglobin and carboxyhemoglobin. When the hemoglobin binds with the oxygen, it is
Oxyhemoglobin.
oxyhemoglobin. Okay, so the prosthetic group, these two, they compete for the same binding site. The carbon monoxide and the oxygen, they compete for the iron or the heme group. But the carbon monoxide always wins because the carbon monoxide has a higher affinity. Now the carbon dioxide, where does it bind? To the ends of the polypeptide chains in the hemoglobin, it binds with the amine groups. So the binding site is not the same. The hemoglobin also binds with carbon dioxide, but it has the least affinity to the carbon dioxide. And where is the binding site of the carbon dioxide? It binds with the terminal amine groups of the hemoglobin. The hemoglobin binds to all of these molecules. It binds to carbon monoxide and to oxygen and to carbon dioxide. Every hemoglobin molecule binds to 4 of those each time. So the hemoglobin can bind with 4 carbon monoxide or 4 oxygen or 4 carbon dioxide molecules. Okay? Are you all with me?
Yes.
Again, so the hemoglobin. The hemoglobin is a globular protein, very important. We studied it in detail. It is made up of 2 alpha globins and 2 beta globins, and it has a central prosthetic group. This central prosthetic group can bind to 2 things: carbon monoxide to form carboxyhemoglobin and oxygen to form oxyhemoglobin. So the hemoglobin binds with the carbon monoxide, oxyhemoglobin, or oxygen to form oxyhemoglobin, and it has a higher affinity to carbon monoxide. And the ends of the hemoglobin, the terminal amine groups, they bind with CO2. But it has the lowest affinity to CO2. Actually, the highest affinity is to the hydrogen ions. This is when the carbon dioxide binds with water. And when the hemoglobin binds with the hydrogen ions, what does it form? Hemoglobinic acid.
Globinic acid.
Remember? So affinity 1, carbon monoxide. Affinity 2, hemoglobinic acid. Affinity 3, oxygen. And the last affinity is carbon dioxide. Good. And we even studied the hemoglobin when it mutates in sickle cell anemia. Good. So this question, going back to it, I'm going to highlight it because they left the equation and the important part, and they asked you about what will happen to the carbon dioxide if it does not react with water. This here, you will say a lot of things. The hemoglobin will bind with the carbon dioxide to give you carbaminohemoglobin. It has the lowest affinity, yes, but it did not react with water. And where exactly does it bind in the hemoglobin? Which part of the hemoglobin does it bind to? I just said it.
الends الامين group.
Yes, and every so it binds to the ends or the amine groups of the hemoglobin, and every hemoglobin molecule can bind with 4 carbon dioxide molecules. Now, when I sent you on WhatsApp and I asked you if you have this, where is this? This is not what I want now. Where are you? Yeah, here. Yes, I asked you here. I found it actually in your notes. Every hemoglobin molecule carries 4 carbon dioxide molecules and it binds with the terminal amine group. So I definitely said it in class quickly because everything in your notes is in the PowerPoint. Okay, so that's why I tell you also again, focus on the details because they can leave the big picture and they can focus on the small things. When hemoglobin associates or binds with oxygen, it forms oxyhemoglobin. State the precise site in the mammalian body where the hemoglobin molecules bind with oxygen. No, that's easy. Where in your body? In your body part, huh? Does the oxygen bind with hemoglobin?
The
Don't
lungs.
tell me red blood cells.
At the lungs.
Yes, at the lungs, at the alveoli. Okay, graphs. This compound is produced in red blood cells, and this compound binds to hemoglobin and it will stabilize it. Figure 3.2 shows oxygen dissociation curves when the red blood cells have high and low concentrations of this compound. Now, obviously, when you have a high concentration of this compound, what does it do to the curve? The curve goes up or down? Why aren't you answering? You're simply telling me what you see. When
What?
this compound binds to the red blood cell, what does it do to the curve?
Miss, can you say again عشان الصوت بيقطع؟
Excuse me?
Can you please say again عشان
When this compound binds to the hemoglobin, what does it do to the curve?
it decrease it
It
goes down.
decreases, it moves down. Yes, it moves down. So when it moves down or it moves to the right, how will this affect the affinity of hemoglobin? More affinity or less affinity?
Less affinity. لا
Yes.
more affinity.
يا حبيبي, yes, it's going down, less affinity. Noor, you have to study. You're not studying. You depend on your memory. You're solving without studying. There's something wrong. Okay, P50 is the partial pressure of oxygen when hemoglobin is 50% saturated. And the P50 is used to compare the affinity of the hemoglobin for oxygen under different conditions. Use the information in figure 3.2 to describe the effect of an increase in the concentration of this substance on the oxygen dissociation curve. We just said it. When we increase this compound, what will this do to the oxygen dissociation curve and how does it affect affinity? By the way, the question is very easy. I increased 2,3-D Pg, high concentration of that. How will this affect affinity? What will this do to the curve? Just give me information from the graph.
The curve will move to the right because the affinity of the hemoglobin to oxygen will decrease.
Thank you. Thank you so much. If I want numbers from the graph, which percentage saturation of hemoglobin will I use?
100%? The maximum Vmax.
No. The Vmax, they never meet actually. You will use the 50%. P50 is the partial pressure of oxygen when the hemoglobin is 50% saturated. It is used to compare the affinity. Read the questions. So I will go through the percentage saturation of hemoglobin at 50 and I will compare. When there is low concentration of this, the partial pressure of oxygen was around 2 point something. And when the percentage saturation of hemoglobin was 50%, here I had more oxygen. If you want to get numbers from the graph. Okay? So what you said is correct. Just add numbers from the graph when the percentage saturation of hemoglobin with oxygen is 50%. 50%, it will be holding less oxygen. So when the 50% of the hemoglobin is occupied in the high concentration of the compound, it will be holding less oxygen. Clear? It will be holding less oxygen because it will reduce the affinity. Are you with me?
Yes.
Okay. In other words, you need more partial pressure of oxygen so that it will occupy 50%. This is tricky. In order to occupy 50% of the hemoglobin, you need more oxygen to occupy 50% of the hemoglobin. If the numbers are confusing you, don't use numbers. But what Mariam said is enough because it's only 2 marks. Are you with me? Okay. The blood is stored in blood banks for use in hospitals during operations. The concentration of this compound in red blood cells decreases when blood is stored in a blood bank. The concentration decreases. State and explain the effect that the use of the blood taken from a blood bank has on the supply of oxygen to the tissues. So when we take the blood from a blood bank, this compound will have less concentration. Why is this an advantage? Why are you silent? This makes me feel that you do not understand the question. Let's go back to the beginning of the question. They said that there is a compound, 23DPG. This compound, when they added to the red blood cells, the red blood cell, they have a lower affinity because the graph shows you that there is a high concentration of this compound and the affinity decreases. So this compound is not good. I don't want it. When I remove it, there will be more affinity. Do you understand the graph? Do you all understand the graph?
Yes.
Yes.
Okay. And then the second part of the question, they said the blood will be stored in a blood bank. And when we take blood from blood banks, it's an advantage. Why? Because the concentration of this compound will decrease. And this is good. Why do we want the concentration of this compound to decrease?
To increase the affinity of hemoglobin to oxygen.
Exactly. Because the tissues in an operation, you have this patient needs oxygen. I want to give him oxygen. So I will give him hemoglobin with high affinity. That's it. So can you see how the answer is so simple? They are twisting the questions. The questions are made to confuse you. Fine. So you will say that the patient during an operation, he does not have enough oxygen and he needs oxygen. So they will give him hemoglobin with high affinity of oxygen when they give him this red blood cell that is stored in blood banks. Okay. Figure 4.1 is a diagram showing the transcription of part of a gene that codes for a collagen polypeptide. And the part of this gene shown is a section of an exon. The exon is the coding or the non-coding part.
Coding.
Coding. Structure A represents an enzyme involved in transcription. What is A?
Metaphase.
You know something? I'm going to close and I'm going to go sleep. What did you say, Kareem? Who said that?
No, it's just I made
حرام
a
عليك يا
mistake.
كريم. لا كريم, this is I'm going to have a heartache or a heart attack. Metaphase, we're talking about mitosis حبيبي. We're talking about protein synthesis. This is something so different. What is A?
DNA
RNA
polymerase.
polymerase.
RNA polymerase, Mariam, not DNA polymerase. I am here transcription. So I am creating a messenger RNA. So what is A?
RNA polymerase.
Yes, not metaphase. Okay. Name the bond that forms between the nucleotides and the primary transcript. What is the bond here?
Hydrogen.
No,
Phosphodiester.
They did not ask about the bonds between the bases, Mariam. The bond in the sugar phosphate backbone between the nucleotides. Noor, what did you say?
Phosphodiester.
Correct. The phosphodiester bond in the sugar phosphate backbone. Okay, state the number of amino acids that are coded for by the sequence of nucleotides on the primary transcript. Okay, this is a tricky question. How many bases are needed for one amino acid?
You showed the figure, yeah.
3. So let's count how many bases do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. I cannot say 16, 15 divided by 3. So what's the answer?
5.
5. Wait, let me see. This is which number? 1 minute. This is number 4. Maybe we are making up biology.
Miss, sorry, I have to go get something fast. Okay.
Okay, اتفضل. 5. Yes, we are right. Okay, so this is 5. Use the information in figure 4.1 to explain why only one of the strands is transcribed and not the other. Why only one of the strands are transcribed, not the other strand? Now, this is also critical thinking. Do these strands have the same sequence of bases?
No.
No. So they don't have the same sequence of bases. If I have a different sequence of bases, this is going to give me the same protein or a different protein. Different protein. So because these strands, they don't have the same sequence of bases. Yes, they are complementary, they're not the same. So I need at least 2 reasons, because I will say that the strands, they don't have the same base sequence. And a different base sequence will give me a different what?
Polypeptide chain.
Yes, a different polypeptide chain and a different protein with a different function. Also,
Miss,
the messenger RNA, yes.
can I say that the translation would waste energy?
If I translate the wrong polypeptide strand, if I translate the wrong DNA strand, yes. So I want to translate the correct strand, the strand with the correct sequence of bases. If I translate something else, yes, I will waste energy. I can write this down. If I don't translate the correct strand, I will waste energy and resources. Also, the messenger RNA must bind to the 3' end to build the new strand in the 5' to 3' direction. So it must bind also to one of the ends, not both ends. Must bind to the 3' end. This is a nice question. Okay. I'll also highlight that because questions can be repetitive, by the way. State 2 features of mitochondria that are visible. What is visible
Double
here?
membrane.
Okay, very good. Double membrane. What else? What are these?
Inner foldings.
Yes, the crista or the inner foldings. Good. EA. The line on figure 4.2 shows the diameter. Now, we will not be able to solve that because you need to measure the line and then convert it to millimeters. Okay. So I will not be able to do this here. Outline the ways in which ions can move into mitochondria. Ions. How can ions move across any
Facilitated
phospholipid?
diffusion.
Excuse me, my dear. It will move by what?
Facilitated diffusion.
You mean facilitated diffusion. Okay, but again, what is the structure that will help ions move across the membrane?
Active transport.
Okay, your answer is just not very organized. Again, what is the structure that will help it move across the cell membrane?
Carrier proteins and
Yes,
transport.
channel proteins. We have 2 types of proteins in the cell membrane, channel proteins and carrier. And what's the difference between them? The channel protein will do active transport or facilitated diffusion.
Facilitated diffusion.
Yes. So please just organize your answer. You all gave me the answers, but it was messy. Channel protein for facilitated diffusion. In other words, down a concentration gradient, but no energy. No energy here. What about the carrier protein?
They use energy to change shape to transform
Of
to
course.
transport ions.
Yes, so it will change shape here against concentration gradient, and I need energy. So this is a nice question. Ions, they cannot move because the phospholipid bilayer, it has a central hydrophobic core, and the ions cannot move because the ions are polar. So you can start by saying that. Why can they move? And they need helping. They need help. So they need either channel protein or carrier protein for the ions to move so that they can cross the hydrophobic core, because these proteins, they have a hydrophilic core. Okay, you have a lot of things to say. The exam is finished? No. Explain why a root can be a source and a sink. Now, this is O level. Why can a root be a source and a sink? When is it a source and when is it a sink?
It's a source when the leaves are not present during winter or fall.
Yes, correct. Okay, so remind me to send you the O level page for the source and the sink because this is O level. So it will be a source when it will be the supply of the sucrose and amino acids. Like Mariam said, when there are no leaves in the winter,
السينك
no
مش
leaves
هيكون
to do
عشان storage صح؟
photosynthesis. Excuse me?
Sink will be for storage.
Yes, it's a sink, very good. When it will store the sucrose and amino acids, or it's going to use it for growth. So in different seasons of the year. Okay? State two examples of assimilates that are transported in the phloem.
Amino acid and sucrose.
Excellent. Again, this is all of very nice. Sucrose and amino acids. Describe and explain. I don't like these questions. The mechanism that is responsible for the movement of the phloem sat in the sieve tubes. This is definitely out of 4 marks. Okay. So you will describe and explain the mechanism that is responsible for the movement of the phloem in the sieve tubes. Yes.
يعني I don't know where to start يعني from where to start يعني.
You don't know from where to start. It's a big paragraph and you don't know from where to start. That is why we have to practice solving exams and actually get a paper and write the answer on the paper, because here you are not only practicing, you're not only memorizing, but you're writing it down. You will start by saying what? That the movement of the assimilates inside the phloem sap, there is a mechanism. Forget the loading and the unloading now. The movement of the material either inside the xylem or the phloem or even arteries. It's called the mass.
Mass flow.
Correct. So you will start by saying that the movement is by the mass flow. Now, then you will continue by saying the reasons why this mass flow is generated. Why does the assimilates move from the source to the sink? What is the force that is driving the assimilates from the source to the sink?
Hydrostatic pressure.
Exactly. So you will discuss the changes in hydrostatic pressure. There is a big story here. Remember, first we have the loading of sugar by the companion cell from the cell wall of the companion cell through the cell membrane with the help of the hydrogen pump and the co-transporter protein. This is 4 marks. So I don't think that they want this part in detail, but you can outline it. Like first, the source or the sugar will be loaded into the companion cell with the help of the hydrogen pump and the co-transporter protein. And then the sugar will move through the what? Into the phloem. It will cross the what? What's this opening called?
Plasmodesmata.
Correct. And it will diffuse from the cytoplasm of the companion cell into the source. And what will happen to the water potential at the source?
Decrease.
Decrease. And this is going to drive water from the nearby
Xylem tissue.
continue. So this will increase the hydrostatic pressure at the source. Now, what is happening at the sink?
Hydrostatic pressure decreases.
Exactly, due to the unloading of sugar. And this change in hydrostatic pressure will create the movement of the assimilates from the source to the sink or the mass flow. And this direction, is it always in one direction or the direction changes?
One direction.
In the phloem.
Direction.
Okay, 2 directions. It's always from the source to the sink. The source can be the leaves up to down, or from the roots to the leaves down to up. So the direction up and down. So here you will write the whole story of the loading of sugar and the change in hydrostatic pressure for the movement of the assimilates within the phloem. And you will focus on the movement of the assimilates within the phloem, not the loading and the unloading of sugar. This is usually one question We solved this question before in the classified, and the movement of the assimilates in the phloem, the mass flow due to changes in hydrostatic pressure is another question. Okay, so you will discuss the changes in hydrostatic pressure here. You're all with me? This is an easy question. I'm not going to highlight it. It is a direct question that depends directly on how much you studied. Okay, what is this? I want to jump, but I should not. Now, the virus infects many plants, and the virus passes between cells. And the enzyme, there's an enzyme called the pectin methyl, I don't know what enzyme, is involved with the production of cell walls. The enzyme is also known to influence the movement of the virus. Scientists investigated, so I should underline that. Scientists investigated the effect of the enzyme on the transport of the virus through the plants. This is the most important sentence. Scientists use 3 varieties of plants, 2 varieties V1 and V2. They have a small quantity of the enzyme, and a third variety, C, has the normal quantity, so C definitely is the control. The plants in each group were infected with the virus, and then the accumulation of the virus particles transported to the leaves at the top of the plant was determined after 36 days. The results are shown, and the arrow indicates when all the plants were infected with the virus. So this is the time they were all infected here. And then the accumulation of the virus, they all show the same graph to me. There is a steep increase and then a steeper increase. But the difference is when it happened. When the virus started to accumulate. Now, before I move on, which one took the shortest time for the accumulation of the virus, the control or V1 or V2?
V1.
ليه يا حبيبي? Which one took the shortest time? V1 started at day 9 or day 7. The increase in the virus started to accumulate at D7. In the control, the virus started to accumulate from the first minute.
2. Yes.
So the
2
V2
V2.
took the
Yes.
longest time. So that V2 took the longest time for the virus to start to accumulate. So let's look. The V2 has small quantities of the enzyme. So V2 has small quantities of the enzyme. So this enzyme helps in accumulation or does not help in accumulation of the virus?
Helps on accumulation.
It helps. You just give me a second. Okay, I'm back. I hope you understand the graphs. When the control had lots of the enzyme, so the control here has high amounts of enzyme, this enzyme helped in the accumulation of the virus. The virus accumulated from the first minute, from day 1. Okay, compare the results for varieties V1 and V2 and the control. Now this question is only compare, only. You will not compare and explain, so it's got nothing to do with how much you studied. It's got nothing to do with anything you studied, actually. You will just compare. So you will write numbers for the control and the V1 and the V2, and you write the conclusion, and you will write the similarity between them and the differences between them. What will you say, in short?
Both V1 and V2 reach maximum accumulation.
Okay, but which one took more time? Which one took less time?
V2 took less time.
Yes, you are right, but you have to be more precise. How much time did the V2 take, and how much time did the V1 take?
V2 took 2 days.
Okay, yes, and you can say that V2 started to show an increase from after 22 days, and V1 started to show an increase after 7
9
days.
days.
You will simply compare. You will simply compare and give numbers. And the control took the least time, and all of them showed a steep increase, and all of them started to plateau after a certain accumulation. Okay, so you will simply compare. I don't even need to look. Let's just look at the mark scheme. This is which number? Mm, we said this. Well, maximum accumulation is delayed. Yes, we said V1 and V2 will take more time. Data to support rate at which accumulation increases. V1 and V2 and the control reach the maximum accumulation. Low quantities of enzyme does not decrease maximum accumulation, or the enzyme quantity is not required to reach the maximum accumulation. Any valid point for any valid comparison. You will simply compare and give numbers and take every graph separately and discuss it. The last question. Explain why the ventricles have thicker walls than the atria. Now, this is an O-level question. Why? Why are the
They pump
ventricles thicker?
blood at higher pressure.
Yes, of course, they need to pump blood at a higher pressure to overcome
And
more
it needs
resistance.
to
Why?
be, the pressure needs to be high so it can reach all the body.
Very good.
How the
Okay,
target is
either
working.
okay, the atria, they have thinner walls because they only pump blood to the lower ventricles, shorter distance, longer distance. You can say that the ventricle, the left and the right one pumps to the lungs, one to the systemic circulation. Very easy question. Okay, now the what initiates the heartbeat?
Sinoatrial node.
Correct. What will cause a delay in the wave of depolarization? After the sinoatrial node initiates the heartbeat, what will contract? Both atria, and then this wave of depolarization will go first to what to cause a delay. The last question.
Anteroventricular
ABN?
node.
Yes,
ABN.
atrioventricular node. Okay. Now, when I looked at the mic scheme, they said something here very strange. Atrioventricular now node and non-conducting tissue. Okay, this non-conducting tissue is a tissue called annulus fibrosus that is in one of the pictures in your notes that I showed to you here, annulus fibrosus. This is non-conducting tissue that is found between the atria and the ventricles, and it also contributes to the delay of the wave of excitation. So the wave of excitation, it is delayed by the atrioventricular node and this annulus fibrosus. You are not requested to know its name, but in the mark scheme I saw it says delay of the tissue, delay of the impulses by the atrioventricular node and non-conducting tissue, just non-conducting tissue. In case you
So
are
the answer
curious
is
and
both.
you,
I should
the
write both.
answer in the mark scheme, don't write annulus fibrosus, just atrioventricular node. Maybe just mention that it is non-conducting tissue or with non-conducting tissue around it.
Okay,
I saw
if
this
I write
answer.
if I
If you
write
write
ABN
annulus
بس.
fibrosis طبعا كويس.
طب
If you
AVN
write, it should be right. دي أول
Yes,
مرة
يعني
أشوف
المفروض نكتب
فكرة non-conducting
two.
tissue.
We should
Oh,
write the two.
yes, as long as and it is underlined, then they want the non-conducting tissue. Or an alternative wording that the atrioventricular node is in between the atria and the ventricles. They wanted this. I don't know. It's the first time for me to see it. This non-conducting tissue, because if we want to explain it now, this non-conducting tissue is called annulus fibrosus, and it is this labeled blue line. This picture is in your notes. And it is in between the atria and the ventricles. So they both contribute to the delay. The atrioventricular node, this small atrioventricular node, and the annulus fibrosus, which is non-conducting tissue located between the atria and the ventricles. Okay? Actually, this was in the old syllabus and it was removed. So I'm just adding it again. Clear?
Yes.
And so atrioventricular node and the annulus fibrosus or non-conducting tissue transmits the wave of depolarization through the muscles of the ventricle. Who will take it down to the muscles of the ventricle?
Protein fibers.
Yes, the Purkinje fibers. And what closes when the left ventricle contracts?
Left anteroventricular.
Yes, which is what? The atrioventricular valve on the left side. You know its name now. The mitral. So the atrioventricular valve will do, the left atrioventricular valve will do, or the mitral valve will do. Okay, we finished this exam. Finally, we finished it. Okay, now let us look at the tricky questions. This question, I don't know why I highlighted. It's not tricky, we're just comparing. Most of the questions were direct, straightforward in this question. This question was tricky. Go over it again, please. Why would it transcribe one strand and not the other? We said it because the sequence of bases are different. So we do not want a different protein. We want a specific protein. We discussed this question. This was not difficult. The hemoglobin, I want you to revise everything about the hemoglobin and what it binds to. I think we explained it clearly. The heme groups, they bind to carbon monoxide and oxygen. It has a higher affinity to the carbon monoxide. And the amine groups, the terminal amine groups, they bind to the carbon dioxide. Okay. So the exam was not difficult. It was relatively easy with a few tricky questions. I'm going to save this sketch and send it for you so that you can go over the tricky questions. Okay? Any questions before I go?
No, miss,
Hello?
thank you.
What is today? Today is Monday. Do you want on Wednesday هيبقى عيد ناخد درس في العيد ولا لا؟
لا.
Lambs.
طيب ماشي. We'll meet you after Eid. Enjoy. No, don't enjoy. Go study.
Okay.
I will go enjoy studying.
You're sure? I don't know why I have this feeling that you still need to study harder. You did not study well.
لا ما انا هذاكر اجمل.
Okay, please. Okay.
Yes.
Bye bye.
Bye.
Bye, miss.